Bioinfocongress IV, Assistant Professor Dr. Fikri Birey, in Vitro modeling of human cortical assembly in health disease. Assistant Professor Dr. Fikri Birey, as a Fulbright Scholar, earned his Bachelor of Biology degree in Biology and Philosophy from the University of Kansas in 2008. He earned his PhD in Genetics on neuroglial interactions in stress-related disorders at Stony Brook University in 2014. He completed his postdoctoral studies at Stanford University in Sergei Pasca's laboratory between 2015 and 2021. Furthermore, he developed the Assembloid platform, a stem cell-based developmental forebrain model, and used it to understand how neurodevelopmental disorders occur during human cortical development. In early 2022, Dr. Birey established his center in the Department of Human Genetics at Emory University, where he continues to produce and use stem cell-based models to understand the guiding principles of how the human brain assembles.
Dear Dr. Fikri Birey and participants, welcome all of you to our Bioinfocongress IV event. Dr. Fikri Birey will give us a talk today on in vitro modeling of human cortical assembly in health and disease. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Fikri Birey, welcome back.
Hello.
If you wish, you can start your presentation.
Sounds great. Thank you so much for that kind intro and inviting me. This is such a fascinating, well-organized initiative, and kind of astounded by the fact that it's all organized by you trainees, students. So kudos to all of you, and again, thank you so much for inviting me. To the end, I also want to thank the people that stuck around all day until this last talk of the day or the night, I guess. But I'll try to keep it short and short and sweet. But what I want to talk to you about today is about our efforts in developing in vitro stem cell-based models of the human brain assembly and how we're using such models to better understand how brain disorders emerge during neurodevelopment. And broadly speaking, I'm interested in the question of how the human brain is constructed during fetal development. And just like many other species, the development of the human brain starts with the formation and closure of the human neural tube, shown here, which in humans happens around 4 weeks post-conception. But unlike many other species, the processes that are triggered afterwards are orchestrated and very tightly orchestrated over very long periods of time. And you can see here in the schematic that some of them really do take place well after birth. For example, things like neurogenesis and migration can take place largely in utero. Prenatally, things like myelination, synaptic pruning, etc., take place largely after birth. Really resulting in this immensely sophisticated and functionally compartmentalized piece of tissue. And this really is an astonishingly complex project. And despite it involving generation and coordination of a stunning number of cells and connections over months, it occurs with an incredible precision and accuracy from individual to individual, which really, I think, highlights how robust the assembly instructions that are encoded in the system are. One of my specific interests lies in the question of how a particular region of the human brain, the cerebral cortex, is assembled. The cerebral cortex arises from these forebrain structures during development, and in adult really performs as higher order cognitive functions, and very interestingly is significantly expanded in volume in the human lineage compared to our primate ancestors. The period during which the first cortical circuits are assembled, which really spans mid to late gestational periods, is thought to be a very critical period during which these principles of mature circuit structure and function are established. And at the cellular level, the assembly of this early cortical circuits really involves two major neuronal subtypes: glutamatergic neurons, as shown in red here, and GABAergic neurons, GABAergic interneurons. And while the glutamatergic neurons are born locally in this dorsal side of the forebrain, which is the part that later gives rise to the actual cerebral cortex, the GABAergic interneurons, on the other hand, born a little bit remotely ventral to this dorsal forebrain side, in the ventral forebrain structures, and in the structures that are sort of transient in development. They're called ganglionic eminences, and one of them is shown here, the MG, the medial ganglionic eminence. So once these interneurons are born in these regions, they really have to migrate, especially in humans, really long distances towards the dorsal forebrain, where they meet their glutamatergic counterparts, and they really start forming these early connections. And that's what I refer to as this early assembly of the cortical circuits. And really the genetic or environmental perturbations to any number of these processes that I just outlined are thought to be linked, very closely linked, to several neurodevelopmental and neuropsychiatric disorders, like autism, schizophrenia, epilepsies. Yet a mechanistic and dynamic understanding of how these processes really unfold in humans and go awry in patients remain virtually non-existent. And this is largely due to the fact that we really lack access to the functional brain tissues, at a level where we can really study these processes dynamically and mechanistically. And this really has been the primary roadblock that my work over the last several years has aimed at address through developing these personalized stem cell-based platforms that can relatively faithfully model these processes in a dish, in a cell culture dish, so that we can really study them directly in patients. But before I go into details about the model, I want to take a step back and really explain the core technology that has enabled us to develop these models in the first place. The starting material for these models that we have developed are called human-induced pluripotent stem cells, or what I'm going to be referring as HIPSCs. These are pluripotent stem cells. And what I mean by pluripotent stem cells is that these are cells that can really differentiate into any kind of body, any kind of cells found in your body. But in this case, they're induced from post-mitotic somatic cells, things like skin cells or blood cells. And this technology was first described by Shinya Yamanaka and colleagues over a decade ago, won a Nobel Prize, I guess, in 2008 or so, if I'm remembering correctly, and really unlocked this huge potential for personalized medicine, really enabling one to have access to any kind of cell type of interest directly from individuals. And I'm going to outline a typical workflow here, which starts by harvesting the somatic cells. These can be skin cells, blood cells, and then through the overexpression of what's called Yamanaka factors, which are these transcription factors that Shinya Yamanaka described, we're able to reprogram these cells so that they assume a pluripotent state. And at this stage, they really look and act like real stem cells. They form these colonies, and given the right cues, chemical cues, they can make cells that come from all three germ layers. And it's in that sense, they're very much like human embryonic stem cells, which are sort of naturally found in the inner cell mass of a blastocyst. And then these cells can actually be dissociated and then reaggregated into these three-dimensional spheroids. And using various patterning molecules, small molecules, growth factors, specific cocktails of these, they can be differentiated into region-specific brain organoids that sort of resemble specific parts of the developing brain. And I'm going to just give you an early example of this kind of work where this human-induced pluripotent stem cells, iPSCs, were aggregated in 3D and really pushed towards certain regional fates of the human brain. And this early work really focused on trying to model this part of the developing human brain, was the dorsal forebrain. As I mentioned to you, this is where the glutamatergic neurons really arise during development. And the work from my postdoctoral lab coined this term human cortical spheroids at the time. And this model really involved generating these 3D structures that were a few millimeters in size, kind of in terms of its size resembled an E12.5 mouse brain, for those of you that come from this kind of work. And when we mapped them transcriptionally to the primary data sets, primary tissue data sets, they really mapped onto the primary fetal cortex that were around mid to late gestational stages. So that was very encouraging at the time. And then we did a lot of characterization showing that they had these cellular features that was reminiscent of the developing cortex. The ventricular zone like structures that had these progenitor pools. They had this sort of lamination like structure to them, where they're the deep layers and the superficial layers, just like how it is in the real cortex. And then they also at later stages went through gliogenic switches, where they would start making this non-neuronal subtypes like astrocytes. But this protocol really lacked a critical subtype. It was the cortical GABAergic interneurons. And this made sense that they were missing this subtype because the patterning strategy really targeted to generate the dorsal forebrain. And you might remember from a few slides back, the cortical interneurons are generated instead in these ventral regions, in the ventral forebrain regions. And then they have to migrate dorsally. So when we started thinking about how to build sort of a fuller model of the developing cortex to incorporate these interneurons, the strategy we took was to separately generate the ventral forebrain spheroids, which we called human subpallispheroids. Subpallium is another name for the ventral forebrain, and sort of assemble these subpallispheroids with the cortical spheroids so that we can start modeling some of these processes like interneural migration and integration onto the excitatory circuits. And we ended up calling these models forebrain assembloids. And we described the first assembloid model in 2017. And after that, several novel assembloid models really followed, modeling various interregional interactions that arise during human brain development. And I'm highlighting a few of them here. And actually going to highlight the latest one. This is a preprint that came out a day or two ago, where we built an assembloid model of the thalamocortical connectivity. This is a hugely essential and fundamental interregional interaction that really drive this early corticogenesis and sort of like this cell extrinsic aspects that depend on the early presensory activity that comes from the thalamus. So this is a very exciting model that we had worked on for a while and just came out. But that really, this is just one example out of many that is out there. So this is like a hugely versatile toolkit now that we have to decipher this principles of human brain assembly. And if you ask me what really makes these models stand out as this robust modeling tools, and likely why we have so many of them out there that came out over the last few years, I would list probably 4 things. That one, that they're modular, that you can make these region-specific organoids separately, and then assemble them at will, giving you really that tight control over when to introduce that connectivity between regions. They're very tractable, unlike an embryonic mouse brain, for example. They're in a dish, and you can really at ease do these longitudinal studies where you can really follow any number of processes over time within a single assembly, right? So that's a huge advantage. They're tunable. Just because they're modular, we're able to sort of introduce various tools pre-assembly to a given region-specific spheroid. And for example, we're able to introduce an opsin only, let's say, in the subpallial derived intraneurons, and then perform the assembly so that you sort of limit the expression of a certain viral vector to a particular region without having to go through like complicated infection schemes or anything like that. So it makes it sort of a tunable model when you think about this aspect. And as I'm going to be showing to you and spending the end part of my talk a lot on this, that they're very amenable to disease modeling. And I particularly worked on neurodevelopmental disorders of monogenic origin, and they're extremely robust models when it comes to understanding those kinds of diseases. But then now people are sort of starting to study neurodegeneration using these other kinds of assembloid models, for example, cortical spinal muscle assembloids. But for the rest of my talk, what I would like to do is to take you back and walk you through the steps of how we built the first assembloid model, the forebrain assembloids, and basically going from the parts of the assembly and how we validated it using primary fetal tissue, which is a hugely important part of any in vitro model. And as I said, just at the end, going a little bit deeper into how we applied this model to really better understand and cover novel insights about a particular neurodevelopmental disorders called Timothy syndrome using patient-derived assembloids. All right. So the first question, as I alluded to, was how do we go about developing the subpallial spheroids? I'm going to be referring to subpallial spheroids as HSS and cortical spheroids HCS. And for that, we started by running a small molecule screen. We took the HCS recipe as our base recipe, and then we basically asked the questions: what are some of the compounds and small molecules that we can use to modulate signaling pathways in a way that we go from the dorsal forebrain to ventral forebrain identity? And we, of course, looked at classical developmental studies that really teased out the interactions between these signaling pathways that really drive specifications of those regions. And we basically focused on the sonic hedgehog signaling, which is very enriched in the ventral side, and the wind signaling, which is sort of enriched at the dorsal sides. So the strategy was very simple. We said, okay, why don't we then block the wind signaling, which gives rise to dorsal identities, and enhance the sonic hedgehog signaling, which is sort of enriched in these ventral regions. And that turned out to be good enough of a strategy. So when we started looking, we saw these progenitor pools that were related to ventral progenitors. And then when we waited a little bit longer, we were able to see these GABAergic interneurons arising. And we wanted to get a full picture of the cellular diversity. And as everybody under the sun does, we did single-cell RNA-seq. And we basically were pleasantly surprised to see initially that our HCS and HSS recipes really segregated. There were the primary clusters that segregated, and within those, we were again happy to see that each of these had their subtypes that we expected to see. And if you focus on the subpolygon, for example, you'll see that there's the GABAergic neurons and the ventral progenitors that give rise to these neurons, as well as some oligodendrocyte progenitors and acerugly as well. And I'm skipping a lot of the characterization here, but once we were convinced that we had a good subpallial spheroid recipe, we went on to assemble these together. And the assembly is quite simple. We just sort of restrict them in space, and then they fuse after a few days. And then we started tracking these intraneurons. That was the first thing we started looking at, the migration of intraneurons from the subpallial to the cortical side. And then we use this virus that is specifically reported that is specifically expressed in intraneurons, DLX12B, GFP vector here. And then in green in GFP, you can see the intraneurons sort of slowly moving in. And after about 24 days, there's like heavy infiltration. And if you sort of 3D construct an assembloid 30 days after fusion, you can see this heavy infiltration of intraneurons. And I just want to take put a side note here and explain the way that these intraneurons move, which is quite stereotypical, that has been described in mouse in great detail, where based on the directional cues that they receive from the environment, this leading branch stabilizes towards the direction of the forward movement. And there's very interesting, this cytoskeletal rearrangements and crosstalks that are triggered, creating a pulling force by the leading branch and a pushing force by the cell rear. And this whole coordination, and then this saltatory mode, so it basically looks like a cell jumping, and this mode of movement is called nucleokinesis. And it's very stereotypical. You recognize it when you see it. So when we started watching these things live, we were happy to see that this is indeed the way that they were moving. And if you take a sort of, I'll give it a second for you to sort of appreciate this. But then I talked about validation being an important step. We really wanted to see if we can compare the kinetics of the movement in the assembloids to the kinetics of the movement from the primary tissue. And for that, we acquired there was some primary fetal cortical sections. We infected with the same reporter, and then we did a very similar kind of live imaging. And then you can see that this is very similar to what we saw in the assembloids. Again, they're moving place to place, spot to spot, using this nucleokinetic events. And if you're not convinced, here's a side-by-side comparison. On the left is an assembloid intraneuron moving, and on the right is a real, quote-unquote, human cortical intraneuron moving. So at that point, we were like, okay, we're able to model this migration phenotype quite well, but are they really sort of following their developmental trajectory, and after migration, stopping, branching out, and really integrating with their dorsal forebrain counterparts, which are the glutamatergic neurons? So to really address these sets of questions, we first started by looking at morphology. And it's sort of a classical thing to look at if you're looking at the maturation of intraneurons, that they go from this bipolar morphology to a more complex dendritic branching. And this is what we quantified first, and there was indeed a significant increase in the complexity, branch complexity of the intraneurons that had migrated and then stopped. And then we asked the really critical question of whether these intraneurons are synaptically integrating to these into these excitatory networks. And then for that, we perform this, wholesale patch clamping experiments on interneurons in 3 different experimental groups. If you look at the little schemes under the bar plots, these are the groups either in subpallial spheroids, interneurons in subpallial spheroids unfused. So this is our unfused control. Interneurons unfused in an assembloid. This is our assembly negative control. And then the actual experimental group is the interneurons that had migrated over. And what we saw in the first bar plot here, you can see that only the cells that have moved over and stopped and integrated are really receiving this excitatory postsynaptic currents from the glutamatergic network around them. So that was really key for us to be able to see that. But then our collaborator did the reverse experiment and asked, okay, are the excitatory glutamatergic cells receiving inhibitory, the GABAergic input? And that wasn't the case. When he patched unfused glutamatergic cells from an HCS, he didn't see any inhibitory this time, inhibitory postsynaptic currents. But then that he saw a sizable percentage of the excitatory neurons in an assembloid receiving this inhibitory postsynaptic currents. So there was this in sort of this bidirectional crosstalk synaptically that we observed that told us that, okay, these are probably really sort of integrating and synaptically connecting, which is sort of what you would expect as the next step in their developmental journey. And then at that point on, interneurons really go through this activity-dependent maturation and subtype specification. A lot of interesting things that they do once they start receiving this excitatory input. And one of the questions we asked is whether they are more mature. And as a proxy to that, we looked at the action potential generation. And I mean, lo and behold, that's what we saw, that the cells that actually migrated over and integrated, they're firing twice as many action potentials as their non-migrated counterparts. Okay, so at this point, I'm skipping a lot of the control experiments and some of the other experiments, but after seeing like the results of these key experiments, we thought, okay, we do have a model that we could model various aspects of this human cortical development with, and this is the migration, maturation, network integration. And we thought, okay, maybe we can now use this model really to uncover some new insights about disease, and really sort of fulfill the promise of a model like this. And we sort of got really interested in this particular syndrome when I was in the Pascal lab. Is this Timothy syndrome. So I'll give you a brief overview of what the syndrome really is. This is a rare, highly penetrant neurodevelopmental disorder, and penetrant meaning once you have the mutation, the percentage of the patients individuals that have the mutation, an overwhelming majority of them will actually present with clinical symptoms. That's what highly penetrant means here. And it's very rare. It's a very deleterious sort of mutation, so it's not found all over the place, but it's a very sort of rare, very sort of severe neurodevelopmental disorder that causes various developmental defects, cardiac defects. But sort of molecularly speaking, it's caused by this gain of function mutation in the gene called CACNA1C, which encodes for particular subunit of a calcium channel. And what it does once you have this mutation, it delays the inactivation of this calcium channel. And this calcium channel is a voltage-gated channel. So it means that the cell depolarizes by incoming activity, it opens, but calcium levels are very tightly regulated in the cells. So it needs to really shut down really quickly so that it can buffer the incoming influx of calcium in healthy cells. Once you have this mutation, it takes the channel really long to really close, and we can really demonstrate this core phenotype using two different strategies. We can do calcium current recordings, and then you can see on the left side that in cyan, the delayed inactivation phenotype is very obvious. That this delay, once we're recording the calcium currents, you can see that delay in the closing. And then we can also look at that by depolarizing the cells using high potassium chloride and doing calcium imaging with that. So we thought this is a good candidate for this kind of model because calcium, among many things that it does, it's been shown to be important for motility, axon pathfinding, these early developmental things that really is related to cells finding their way in the developing brain. So basically, we had human iPSCs from 3 different Timothy syndrome subjects, and along with 8 control subjects, we generated this set of assembloids, and looked at how these cells were moving, basically. And initially, what we noticed that the Timothy syndrome intraneurons were moving very inefficiently. And when we try to tease apart what this phenotype might be, sort of in terms of its components, we noticed that the Timothy syndrome intraneurons were moving more, they were jumping more often, but the length of their jumps were smaller. And one thing that is sort of cool and is related to one of the advantages that I listed, the modularity, one thing that you can do, you can mix and match genotypes. So you can put, in this case, TS subpallium with a control, a TS HSS with a control HCS. And what that enables you to do is ask this cell autonomous or regional autonomous versus non-autonomous question. And in this case, what we show that even though we had a control cortex, having a TS HSS was enough for us to see the phenotype. And then we sort of thought maybe we could pharmacologically rescue this phenotype. And for that, we used a drug called nimodipine, which blocks the L-type, this kinds of calcium channels. And the rationale was if the delayed inactivation is letting a lot of calcium in, maybe by blocking a little bit this channel, we can ease the burden on the cells coming from that extra calcium. And that was a very robust rescue for a part of the phenotype. So the saltation length phenotype was really robustly rescued, but astonishingly, it didn't do anything to the saltation frequency phenotype. So this was such a puzzling thing for me that it made, basically I spent the second half of my postdoc trying to figure out why this is the case. And it really took us down a rabbit hole where we ended up really teasing apart the molecular drivers of these two components of this migration phenotype. And I want to spend some time going through the details of that. And initially, when we were thinking how to go about really understanding what is going on, we thought, okay, one thing we could do is really watch these cells closely, really look sort of phenomenologically at the movements, at the morphological changes as they were migrating, and to be able to really track subcellular compartments. We use this algorithm called deep lab cut that people use for actually tracking, markerless tracking of animal body parts in behavioral studies. But we sort of adapted it for intraneuron live imaging, and we were able to reliably track different sections of a migrating intraneuron, and just focusing on the front and the rear of the soma, which are sort of as a readout of the nucleokinesis event itself, and as a readout of the the pushing and the pulling forces, we were able to identify this very interesting phenotype that although in controls, the front and the rear move together, in the Timothy syndrome, the front moved first, and then minutes after, in some cells even, the rear followed. So almost there was a uncoupling of the coordination between the pushing and the pulling forces, and then we quantified that by the correlation coefficient between the rear and the front movement. And once we knew that there was a cell contractility phenotype, we immediately honed in on this signaling pathway, which is the actomyosin contractility pathway. This is the pathway that is involved in making your heart contract, and it is heavily calcium dependent. So it really made sense for us that this pathway was probably overwhelmed in TS by this extra calcium that was being brought into the cells due to the mutation in the channel. So we did a very similar thing in terms of sort of pharmacological rescue of this phenotype, and then we thought, okay, what if we block some of this pathway again that is phosphorylated through calcium, and if we sort of block the kinase that does the phosphorylation, maybe again we ease the burden on this pathway a little bit. And again, this worked really well in the saltation length. Again, it did nothing with the saltation frequency. And this is kind of like a gnawing puzzle for us. It's just like, why? Okay, we sort of figured out the contractility deficit is completely separated from overall motility of the cells. And basically, to be able to figure out what is going on, we went back and did an unbiased RNA sequencing experiment from various different time points, from the HSS, from different patients, controls. And I'll cut a long story short here and just put a one plot to really summarize the relevant finding. Was that we in this age where we're doing a lot of our migration assays, we saw the top two terms that were so upregulated in Timothy syndrome HSS were the regulation of membrane potential and the synaptic transmission, GABAergic synaptic transmission. And here I'm going to pull up some of the subunits that are upregulated in TS that are sort of contributing to this term. So that was interesting. This is the GABA receptors expressed by a cell type that is actually synthesizing GABA itself. So this pointed to a potential autocrine signaling that might sort of potentially be related to this saltation frequency phenotype through the action of the GABA, which was shown to be pro-motility before in other various studies. But before that, we really wanted to sort of functionally validate some of these findings that we saw in RNA-seq. And for that, we went and actually in migrating interneurons, put in a patch clamping pipette and really wanted to record this the membrane potential in actively migrating interneurons. And we really did see a robust depolarized resting membrane potential phenotype in the interneurons. And then that sort of like made us ask whether, this maybe is making the cells a little bit more prone to be active, and then to assay that with the calcium imaging in interneurons, TS versus control. And we indeed see that Timothy syndrome interneurons were sort of had an increased number of calcium transients. And yeah, so this was very interesting, but then we were scratching our head about what might be really the reason why we're getting this increased this hyperactivity. But then it occurred to us that it really could be GABA itself and nothing else, because GABA is depolarizing. As you might know GABA as an inhibitory signal, but GABA is really during development due to the fact that these two chloride transporters are expressed relative to each other. it makes the relative expression of these transporters makes GABA excitatory mostly during development. And that was also the case for our interneurons when we did calcium imaging in these interneurons and added some GABA, we really saw a response. But to really sort of pinpoint the fact that, yes, adding GABA in these interneurons does create an inward current, but not only that, the TS interneurons are more sensitive to such a GABA treatment. We did this thing called the GABA puffing coupled with GABA current measurements. And to cut a long story short, in Timothy syndrome interneurons, we really indeed saw this enriched inward GABA current as a response to GABA puffing. And you know, this sort of like tied in the loop for us a little bit that, yeah, so there's probably this GABAergic signaling deficit due to the fact that there's GABA receptor upregulation in the cells. So once they're secreting GABA, just as they do, as they're migrating, it is going back and really signaling through these receptors that are upregulated in the cells. And whether this could be really the reason why these cells are sort of like higher in their motility as sort of assayed by the saltation frequency. And when we use a similar pharmacological technique where we sort of antagonize some of these GABA receptors, and then we saw that we're indeed getting this rescue of the saltation frequency, but this time nothing was happening with the saltation length. So, you know, I went through this sort of detailed molecular mechanisms related to this interneural migration phenotype, and I'll just summarize what we found here. Was that in interneurons that are migrating, there are two independent pathways that are dysregulated that we think really drive different aspects of this migration phenotype. One is the actomyosin complex that we think are more acutely dysfunctional, and then we have this autocrine dysfunction that is more indirectly dysfunctional because we think the GABA upregulation of the GABA receptors really is the result of this activity-dependent pathways that are being sort of hyper-driven by this incoming calcium influx in Timothy syndrome. And I just want to, you know, wrap up this story here real quick, and just to point out that, you know, I feel like this whole story, I mean, not only points to the fact that these L-type calcium channels are really central to many aspects of cortical assembly, but I really want to underscore the nuance and the breadth of new biological insights one can gather about disease using this patient-derived assembloid models. If one were to just do a simple migration assay, for example, maybe you might be able to get an elementary level insights into what might be going on, but then this model really allows you to really tease apart at that molecular level what the drivers might be, and that could be really essential if you really want to target very specific pathways towards therapies that, for example, you know, this sort of underscores how important GABA signaling is pre-circuit assembly, and if you want to sort of avoid this actomyosin pathway towards restoration of the migration in these patients, you might instead go for the GABA signaling modulators instead of touching the actomyosin signaling, which is again important for the heart. And I'm not gonna have time to really go into the details of some of the follow-up studies that we're doing. There's a very exciting study now that, based on the insights that we've gathered, we're moving towards more of a therapeutic study where we are thinking of ways of genetically modulating this channel in an in vivo setting, and then whether that could sort of be more sort of a clinically relevant going forward for this particular disease. But in my lab, what we're really interested in is really understanding what the molecular drivers are in the spirit of what I described in Timothy's drum in the second part, we're sort of moving towards this idea that what are the molecular drivers that really drive this assembly of the human cortex, and how can we study these using these assembloid models. And I'll just highlight a few directions now that we're actively pursuing. Again, we're really interested in activity and how it really drives this cell extrinsic mechanisms. We are fascinated by interneurons. If it has not been clear so far, we are really thinking a lot about their developmental trajectories and how the many interneuron subtypes now that have been described, how these diversifications come to be, and what role activity, this presensory activity, plays in their development. One thing I didn't talk about was isoform shifting. The CACNA1C, the gene that causes the TS, once mutated, goes through these really interesting isoform changes in development. And there's a lot of genes that really do this alternative splicing dynamics. That is a completely black box, and we feel like that our assembloid models are really sort of uniquely well-suited models to be able to study these kinds of dynamic, temporally resolved dynamics that like this shifting isoform landscapes. And we're developing a lot of new assembloid models to study various different interregional interactions. One of them is one thing that I'm really interested in this early neuromodulatory inputs to the cortex, like, for example, serotonin, norepinephrine. These neuromodulators come in very early development, but not much is known about what they really do. And given the nature and the advantages of the models like these, we feel like we can build these models where we have a region-specific an organoid where the serotonin neurons are born, and then we have this at the cortical, and then we can really control when the serotoninergic input arrives, and then we can sort of modulate that input. And we're really interested in that world of neuromodulation and its effects in early development. We're developing tools to study, again, how this early, in the spirit of looking at specification and regionalization, we're building tools and implementing tools to really understand how regional patterning happens during human brain morphogenesis. This is sort of like taking a step back and moving away from the cortex. So we're building a lot of CRISPR-related tools, implementing these tools, and what I'm showing you here is a particularly interesting one where we're hoping to record temporally resolved dynamics of signaling as the cells are maturing and acquiring certain fates. So we're very excited about this particularly new direction in the lab. And again, all of this in service to understanding disease better. And one particular direction now that we're heading towards is this idea of convergence, and really trying to understand how genes that are of various functions, for example, chromatin modelers, ion channels, transcription factors, how all of these genes can really converge to confer risk for various disorders like autism, schizophrenia, etc. And then on the flip side, we're also interested in this question of how can a particular gene, which is what I'm showing here, is a TCF4, depending on the mutations that it accrues, depending on where those mutations land, can really give rise to wildly different disorders. And with that, I'm going to close. Thank you so much for listening, and I would like to thank the former and current members of the Pascal Lab, where I completed most of this work. My lab opened at Emory in early 2022, and we're finally up and running. This is from a picture from late last year. And one last thing that I'm gonna highlight is the Brain Organoid Hub, please visit this website that will tell you more about this initiative that we're building at Emory, that we sort of put our minds and hands together. This my lab, the Anderson Lab and Sloan Lab, which are both organoid experts, and we're sort of building this initiative to standardize and optimize these organoid models. And if you're at all in this space and you have like these burning questions in your mind, we're running experiments, we're forming collaborations, and we're sort of sharing our data with the field in the hopes that we get to a place where this is a widely accepted model system. Yeah, so if please do visit our website if you're interested. And I'll close with saying that we're recruiting at all levels, and if any of the things that I mentioned to you really grabs your interest, please do reach out, and you can find more information on my lab website. I'll end with that. Thank you so much, and happy to take any questions.
Thank you very much for your presentation, Dr. Fikri Birey. If you wish, we can go to the question and answer section. I'm starting with the first question. Could assembling brain organoids that resemble different areas into assembloids be used to model aspects of the interactions that occur between regions in the human brain?
Assembling that resemble be used to model. Yes, I mean that is our ultimate goal, to be able to model exactly this, the interactions that arise between. So we are sort of limited at the moment to these interactions that arise during development. The more sort of nuanced and complicated interactions that the circuits have during the adult, sort of like this postnatal to adult development, the end stages of development, I would say we're not there yet, but we are getting more and more proficient in really building whatever interregional interaction that you might be interested in, and really sort of honing in on like different region, different thalamus and this and the striatum, basal ganglia, and really building these region-specific organoids. We're really getting good at that. So and then you're just left with assembling them, and now we have a lot of tools in our toolkit that we can use to molecularly, cellularly, functionally really probe what's going on.
Thank you for your answer. I continue with the next question. This comes from YouTube. Neural organoids has lack of circuits connectivity that exists in vivo. So how can we build wiring?
Yes, that's a good point. As I said, we are sort of limited, and the way that I refer to wiring is very rudimentary. So it's this early connectivity that involves this level of synaptic integration, but there's also gap junctions, there's extra synaptic communications, and there's no spatial organization to how these things are connected, right? So if you compare it to an adult cortex that is sort of heavily integrated, well, both locally and remotely from the periphery, it looks nothing like that, right? The connectivity that we get. And people have sort of been thinking about this problem, and one solution that is sort of some people are moving towards is transplantation studies, where you can take these organoids and as a hybrid model put them in a mouse, developing a mouse brain, and they sort of thrive and survive, and they really get integrated into the mouse circuitry. So that's as close as we have been able to get to be able to model real sort of a human-like circuitry, I would say.
Thank you for your answer. Next question is, What are the temporal, spatial, and cross-species limitations that preclude the formation of human neural circuits with high fidelity even after transplantation?
Yeah, that's a great question.
Human cortical organoids at early stage in development.
I mean, this is one that I would have to, yeah, because there are certain there are certain things, there are certain features that are unraveled post transplantation. A striking one is how mature these neurons get, and morphologically, electrophysiologically. I think there's still roadblocks when it comes to specification. When we look at cell type specification and how granular, how specified, let's say, interneurons might get, or cell states, even like the microglia and astrocytes are very context dependent when it comes to their cell states. And whether we can faithfully recapitulate that remains to be seen. And yes, the question of species dependency is a good and complicated one. And it's yeah, I would venture to say that there are certain things that would probably be out of our reach for a long time, and we probably won't be able to even get there ever. And this in vivo hybrids is just putting it in a sort of like another mammalian species probably is as close as we're gonna get in terms of introducing an in vivo environment to these things. These are fantastic questions, and I'm sorry I don't have a more convincing answer.
Thank you for your answer. I'm moving with the next question. Are standardized electrophysiological assays sufficient to improve the reproducibility of electrical recording of neural networks in cortical organoids and assembloids?
I'll just read the question 1 second. Yes. Yeah. We have been doing now standard classic patch clamping experiments for a while now, and they've been sort of our go-to when it comes to this physiological characterization of these neurons. They're sufficient to improve reproducibility on neural networks. I'm not sure if I understand the question of reproducibility. There is a level of variability when it comes to maturation states within a given organoid. I mean, if you patch 10 neurons there, you're going to see a gradient of maturation states. And we sometimes use viral vectors to really hone in on a particularly mature population, for example. So we have tools that really can help us with this kind of variability. And then we have tools that we assay networks with, like multi-electrode arrays, extracellular recording tools that maybe don't really have that cellular resolution, but you have the network level resolution. And there's a level of variability there, but if there's a robust phenotype, let's say when you're disease modeling, you're usually able to see a sort of reproducible enough of a phenotype that if your numbers are high enough, then statistically speaking, you'll be able to find your phenotype. But I think this question of reproducibility is a larger one. It goes beyond the question of electrophysiology, and it's something that the field has been sort of struggling with, for lack of a better term. And it is at the moment our best tool toward combating this variability. It has been numbers. So just like a lot of differentiations, a lot of lines, compared to a mouse study, maybe it's like tenfold increase in the number of lines that we use, or like number of times that we try and experiment. But one thing to also keep in mind when it comes to variability reproducibility, that there's real biological information in variability, right? So some level of variability that we see inherently has that biological information that is relevant, and it interacts with the genomic background. So it's a nuanced discussion whether variability that you see is coming from a technical or versus biological sources, and it's something good to keep in mind.
Thank you for your informative answer. The next question is comes from YouTube. Can bioprinting applications offer a solution to the optimization need of 3D multicell systems?
Mm-hmm. Absolutely, yeah. So, bioprinting, we're thinking about bioprinting in a number of different ways, but I'll just highlight a couple of them. So one issue that I sort of like alluded to was this lack of organization, and bioprinting approaches that we're now thinking about is related to introducing cell populations that secrete this what's so-called morphogens, secrete these growth factors or patterning molecules that activate or antagonize signaling pathways towards inducing a certain regional fate. And one of the ideas that we're sort of thinking about more and more is whether we can use bioprinting to really spatially restrict where we put these cells, so that instead of like bath applying some of these molecules, which is how we usually do our patterning, we can instead introduce cells that secrete these molecules in a sort of spatially restricted manner, so that our organoids, every time receiving those signals from very defined spots in space, and then whether that can be used to sort of control the cytoarchitectures spatially a little bit. Others have thought about using bioprinting when it comes, it's more about building scaffolds and putting an organoid and then building a scaffold and bioprinting, for example, other cell types that you really don't get in organoids, like vasculature, microglia, things like that. So I would say it's a thriving sub-branch of our broader field, this bioengineering approaches related to bioprinting.
Thank you for your answer. The next question it comes from YouTube again. Could the assembloids of the future even bring together organoids from different systems, allowing the study of multisystemic diseases in which the immune system comes into contact with the nervous system?
Yeah, that's a fantastic question, and the answer is yes. If I'm understanding the question correctly, this is the peripheral assembloids that where's the cardiac, where's the gut, that you can sort of introduce to this neural organoids. And there's definitely a number of labs that are very interested in this kinds of questions. When it comes to neuroimmune interactions, I think a lot of labs interested in introducing microglia ectopically, sort of generating this. It comes from a different germ layer, so it has to be generated separately, separate from the corticals or brain organoids, and then they're basically transplanted onto the organoid. So people have published and been thinking about these kinds of approaches a lot, and it's not something that we do in the lab, but there's definitely a lot of potential. I would say it's sort of more of a dormant part of the field. I think it will definitely in future be a staple, I feel like, of what we do, this source of assemblies.
Thank you for your answer. I'm moving with the next question. What are the gold standard and popular practices in designing experiments with assembloids?
Yeah, that was also another great question. So we, these standards sort of differ from lab to lab. Just because I come from a disease modeling background, we do have to sort of put in the numbers to be able to say, that there's a meaningful difference between patients and healthy individuals. And it's just as a standard practice, we've been sort of reporting in our supplemental tables precisely how many organoids from how many differentiations, how many iPSC lines, and this, at least that it is out there. There is a certain level of variability when it comes to assay to assay. Again, if you're doing sort of electrophysiology, which is relatively low throughput, you're sort of limited in your numbers. But when we're doing calcium imaging or so that migration imaging is it's a higher throughput assay. So there are practical considerations, but I'm differing from like giving real numbers here, because it will again depends on power analysis and this and that. But you run a little pilot and then you see the robustness of your phenotype, and then you run your power analysis to tell you exactly the sample numbers that you would need to have. I mean, all I can tell you that it does require multiple lines, multiple differentiations, and from each differentiation, multiple organoids or assembloids. But if you can always go back to our papers and see exactly what kinds of numbers I'm talking about.
Thank you for your answer. I'm moving with the next question. This one comes from the YouTube. Recently, lab-grown clumps of human brain cells were transplanted into the brains of newborn rats. Is there any ethical issue for this application, such as humanized animals?
Yeah, I think this is a good question. There's definitely a very, that it is, yeah. So I'm sort of hung up on the word humanize because this is the word. It sort of depends on where you draw the line. Is it in putting one cell of a human neuron into the cortex is humanizing, and if that's also that's not humanizing, why putting an organoid humanizing? And if that isn't the humanizing, what is it actually doing that it's humanizing the animal? Is it making it smarter? Is it making it suffer in a way that a human might suffer because of the increased cognitive capacity? And that's why you're running into an ethical minefield. So I think there are rules and regulations in place that prevent anybody from doing very early transplantations, and that's just a legal requirement that you just have to abide by, and you cannot transplant these things. I don't remember the exact numbers now, at this very early stages where you might say, okay, this is a humanized rat, where like 70-80% of the brain tissue is compromised from human cells. But beyond that, it is a little bit difficult to draw the line and to really know what we mean when we say humanize. And I think this is ultimately a ball of cells still, right? So they are, people have shown that they are integrating into the rat, and the rats are not suffering. So I feel like that is the sort of the end goal of any ethical discussion related to organoids, which is related to any kind of mouse work or rodent work. That is the animal suffering or not? And we have very clear indications, right? Like when an animal suffers, and then we can sort of like work around that. But again, sort of like a non-answer I gave. But yeah, there's a lot of people that are more qualified in this than I am that can give a better answer.
Thank you for your answer. Another question is, do other neurotransmitters secreted by interneurons also play a role in TS migration phenotype?
Yeah, excellent question. This is not something we looked at. It's but yeah, interneurons do secrete other things. There's other sort of upregulated pathways that I have not shown you that could contribute to the TS phenotype. We sort of like picked the top picks, and then when we saw that we were able to get good rescue, that is sort of where we left the study. But that's a completely reasonable question to ask, whether there are other pathways. And honestly, I all I could say is that we have not looked into that. But excellent question.
Thank you for your answer. The next question is, will TS defect lead to changes in network integration and maturation of cortical interneurons?
Yeah. Yeah, I did not, again, a very good question. I did not have time to show you, but we did look at network changes in assembloids. And what we saw was that it's a bit of a complicated question, because the network dynamics prior to the arrival of interneurons are already different just within the glutamatergic population. And then when interneurons come in, being hyperactive and GABA being an excitatory signal, it really drives that hyperactivity even more. So you get this really intense, this global hyperactivity, waves of activity that you don't really see in the controls. And a subset of these patients have epilepsy, and we always get asked whether that's sort of related to any kind of seizure-like activity that you're modeling. It's hard to say, but we do see network level changes, definitely. When we looked at the stages that we looked, we did not see any major maturation defects, but we have unpublished results showing that if we sort of let these interneurons really mature longer than what we reported, we do see differences in maturation, that they are sort of lagging behind a little bit in their maturation state, electrophysiologically speaking. So there are, but the main question for us is that how much of this later integration maturation related phenotypes, how much of those phenotypes are really connected to the migration phenotype itself? And that's a harder question to answer. But yeah, so there are phenotypes all throughout the sort of like this in vitro cortical development.
Thank you so much for informative answers. Lastly, I want to ask that what advice would you give to the students studying in the scientific fields?
I would say read. This is a fairly new field of study. It's a really sort of hot topic to, people that have not been organoid scientists are getting into the field. And so there's an explosion of studies, but nevertheless, it's a relatively young field. And if you put in the time, it's surprising how fast you can catch up with the state of the art. So that is a sort of, practically speaking, an advantage for any of you who might be sort of interested in this kind of work, that I would just go and read and try to sort of grab as much of what's going on as you can. And I would suggest reaching out. That's really, I feel like, when I was applying, when I was trying to, I was born in Cyprus, and then I left Cyprus 2004-ish, and then before that I was trying to find my way, like how can I get connected with people so that I really find out what I really like to do. And people told me then, and people have been telling me all throughout my training over decades now, that you just have to get yourself out there and really just reach out to people. And network is such a critical, essential part of this. But you first need to find your passion. Like you need to find, whether it's this or AI or regeneration or anything else. And I think exposing yourself, finding that thing that really keeps bugging you and exciting you, and you keep going back to that thing. And once you find that thing, really like chasing the people, and people, I think, connect to that. Like if you're really passionate about something and then you reach out to them, they're also passionate about that. They also went through the same set of steps. So once, it's kind of finding a kindred spirit in a sense, that they connect, they click, and whether that's through an email or through a conference or anything like that. So read and connect with people. That would be the two take-home messages I would say.
Thank you for your helpful advices. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this informative presentation.
Thank you so much. It was a pleasure.
I wish you healthy days.